Thanks for the invitation. I'm very happy to be here with you. So the idea is to present the latest guide we did together with Terry Atwood and one of my colleagues. So it will be really concrete examples of what we can do in a classroom. Just for those of you who doesn't know me, I'm working at the Swiss Institute of Bioinformatics, and we are involved since 2000 in events and games and producing website and workshop for the public at large, and especially for the classroom. So in a normal year without COVID, we get between 2,000 and 3,000 participants for about 60 events. And I would say more than 2/3 are within the classroom. So the idea of what we do with the classroom is always to bring a biological story, a biological context, which is linked to peer review articles. And then we provide pen and pencil approaches to make the people discovering the scientific and the bioinformatic approach. And then we do the same activities together with the help of computer. And this is done with the help of experts in the field each time. And we test the activity within the classroom before doing the guide and the publication. And most of the time, all the guides are focusing on the same databases, such as GenBank, RefSeq, UniProt, or PDB, and are using the same type of bioinformatic tools, BLAST, multiple sequence alignment, Translate, and the genome browser. Of course, sometimes there are specific tools when needed, such for the latest one for the Pangolin COVID lineage assigner. All the activities which have been done by the SIB are on the web page here. It's in French, sorry, so the language is really a major problem for everyone. And as an example, the first activity we did was published in an EMBnet journal. It was about metagenomic and how you can identify species in a mixture, such as the pizza. This workshop is adapted for other type of biological samples, such as the hair, analysis of hair, of a birthday cake, and the last was from a cheese fondue from Laurent Falquet, who tried to analyze the content of a fondue to be sure that it's a Gruyere AOP, which is found in the fondue. And there was a first practical guide done by Teresa Atwood. I was not involved in this, but just to let you know that there is also something about metagenomic. And for those who were at the GOBLET AGM in 2014 in Toronto, we gave a workshop together with the high school teachers and together with Patricia Palagi. And these workshops was finally put into a publication done also together with Patricia Palagi and Terry Atwood, using bioinformatics to understand genetic diseases. And this publication had a great success. We had a lot of view and download. We are happy with that. And more recently, we did a workshop concerning drug design and how we can acquire a simple yet realistic picture of how bioinformatics is used in designing drug today. So this website was published in 2 different journals. Sorry, chemical education, and so we were very happy with that. And again, together with Terry, we were able to do a publication in the GOBLET series, introducing computer-aided drug design. And the last one is the one I'm going to talk more in detail today. So it's to discover the bioinformatics stories related to the new coronavirus. And yeah, because we were learning so much so fast, for me it was very important to propose something where we can digest all the data which were available for the pandemic and to make them available in the classroom. So we did that together with Philippe Lemercier, who is expert in virology, and again, together with Teresa Atwood, who helped a lot in structuring the information. So now the idea just to show some example of what we propose in this guide. So the idea was really, as I said, to digest the massive amount of data which are available to make it usable in the classroom, to have some idea of how you can do some fact checking. This is quite important during this pandemic. So we could, you can, thanks to this guide, discover the genome of SARS-CoV-2, compare genome and look for the variant, identify the variant and the mutations, translate the genomic sequence into the protein sequence, especially for the spike protein, discover the 3D structure of the protein and understand the impact of the mutation on infectivity and immune response. And also, last but not least, we were given an example of how you can infer the origin of SARS-CoV-2 by comparing protein spike sequence. So again, as I said previously, everything is focused on the data which are found in the database, such as GenBank and RefSeq. Then you can compare this data, look for the mutation, translate the data and into a protein sequence, look in the 3D structure, and have a look about the impact of the mutation on the protein structure. So starting from the reference genome of SARS-CoV-2, we first propose an exercise when the people can check the specificity of the PCR test. As you may know, there were quite a lot of discussion about the specificity in the corona septic people. So we provide some example of primer, and the people by hand could have a look at the presence of the primer within the genome of SARS-CoV-2, and then they could check if the same primer were mapping other genomes such as SARS-CoV-2, flu genome, or measles genome. So they can really understand that the primer were highly specific for SARS-CoV-2. So this is done manually, and then they use the genome browser at UCSC. They do a BLAT to check that the primer are found in the SARS-CoV-2 genome, and they look and they find it at the correct position in the SARS-CoV-2 genome. And then they look if the primer are also available in the human genome, and this is not the case, of course, and this is great. So this is an example of what we do. And it's not in the guide, but now when I'm doing the workshop together with the students in the high school, I can go to this Nature publication where they have put the list of all the primer which were used for the PCR test during the pandemics. After that, we just explain how to get the sequence which have been made available on the open data platforms, such as at the NCBI or at the EBI. So how you can, by knowing the accession number, have access to the FASTA format of the sequence, and then you can align the sequence and look for the mutation. And especially we focus on one mutation, which was very important in the alpha variant of the virus at this position in the alignment. So the people have to be, okay, now I've found a mutation at this position, but what does it mean? And how can I learn more about that? So we have, of course, to focus on the protein sequence. And we explain how from a GenBank entry, you can get the protein sequence, and just make also some, gives also some information on the fact that this sequence is the one which is found in the mRNA vaccines. And so this is the link directly to what the people are doing, are living now. So the people can translate the DNA and discover that at this position in the protein sequence of spike, there is a mutation which was called Nelly, which changed an amino acid for another one, and which make the scientists very afraid about that. So why? And just we explain how we can recognize the different variants, which depend on the position of different mutations. So this is to make the things more clearer for everyone to understand what is the variant Delta, what is the variant Alpha, and so on. And then, of course, we talk about the 3D structure of the spike protein. So we have printed in 3D the spike. So we were happy to be able to do that. And to explain that a single protein on the surface of the new coronavirus is responsible for the transmission in human and the resulting pandemic. So just one protein, and we are living for now several months in a strange world. So according to the data which are available, we know which part of the protein is important for the binding to the human receptor, which part of the protein is important for the binding to the antibodies. So the students have to look at the different mutation and to try to locate this mutation in the 3D structure of the protein, and they will see that all the mutations which are here on the list are located in the region which have an impact on the infectivity or on the immune response. So this is why this mutation was part of the variant which are variant of concern. And last but not least, thanks to my colleague Philippe Lemercier, I had access to all the spike protein sequence from different coronaviruses. So we gave access to this sequence and the people could compare them and just, okay, which, and could answer the question, which sequence is more similar to the human SARS-CoV-2 sequence. And in this example, it's really the bat coronavirus sequence, which is much more similar, but the pangolin one is also quite similar to the human one. So that's why the hypothesis was that either a bat or a pangolin or a bat and a pangolin would have been at the origin of the SARS-CoV-2 pandemic. And by aligning all the spike protein sequence here, we get this nice tree, and we could interpret this tree saying that the pandemic in 2003 originate from a civet. The pandemic from 2012, the MERS one, was originate from a bat, and the one we are living now may originate from a pangolin or a bat. So all these figures are in the guide and with some explanation, and just say, beware, this is just an exercise to understand how we can infer the answer of what is the origin of SARS-CoV-2, but of course we need much more sequence to be able to answer the question. And at the end of and even now, the origin of the virus is not firmly established. So we get some nice feedbacks from the teacher. They were very happy. They loved the manual approach before using a bioinformatics tools. They thanked us to digest all the data which were available on SARS-CoV-2. And they say that, yeah, the guide are at the good level for the people in baccalaureate study. So this is just the level they need for the students. So we have now a new website. It's in French yet, but we're going to publish it in English very soon. And we have taken some of the example about SARS-CoV-2, so the people can translate directly online the protein sequence and look at the effect of the mutation on the protein, if there is an effect or not. And also we have explained more in detail the work of NextStrain, so to follow the pandemic with phylogenetic trees. So we just explained the different steps, how we can construct phylogenetic trees for the virus, having at disposition the different genomic sequence. And this was done thanks to the help of Emma Otcroft, which was really involved in this website. And we also now currently have a new workshop on the quest of orthologs, explaining people how we can compare the genome of different species and how we can do that. And there is a funny website where you can create memes by comparing human with different species, and you can discover that we share about 25% of our genes with the banana. We can do it for the cat, the dogs, the mosquito, drosophile, and so on. So the website is not yet on. It will be officially on in November, at the 24th of November, which is the Evolution Day in 2021. But just for you, I just present some of what we are doing now. And maybe, I don't know if Terry agree, we could do a next bioinformatics guide for the high school teacher about the quest for orthologs. Last year already presented the new part in chromosome work, where we did a lot of work about precision medicine. And this is also something which could be found in a practical guide when we explain how we look for the mutation in cancer cells, how we look for the impact of the mutation and which protein is altered, and then how we could add a drug on which could target the protein which is altered. So this could be also a next practical guide if Terry would agree. So I think these guides are really good to bring bioinformatics into the classroom. They unify, they standardize, and they promote the material which is already available about different GOBLET members, so we can reach the high school teacher worldwide. And the question was raised, yeah, the translation is something which is very important. So Celia gave the answer. If you are interested in translating one of these guides, it could be done by sending an email to info@mygoblet.org. And the question is also how to better communicate about this guide. Can we use websites which are focusing on the interaction with high school teachers, such as Scientix or other? I don't know. The question is open. And I will take the opportunity here to thank Terry for her wonderful energy and patience. Thank you really. Thank you so much, Terry. So thanks to all of you for your attention, and happy 10 years anniversary to GOBLET. And if you have any question, you can contact me directly or with the chat or when you want. Thanks.